I think we have people also in the Zoom call. And I would like us to do a little interaction session. If you are in Slido, you should be able to get into the site. Let's see. Hopefully this will work. What institution and department do you work at? Well, this gives us a sense of who we are as a whole community. I see a lot of people from UAB, but I also see people from other institutions, Georgia Tech, University, Kentucky. This is great. Now I'm going to go to the next one. And see what is your current job title or role. Pretty exciting to see that we got many faculty, primary principal investigators, and we're hoping that your labs are also participating and graduate students. We do see about, good proportion of graduate students coming in. All right, let's go to the next one. Okay, I think this is a trivial, so I'm gonna go to this one. So what would you like to accomplish today? Some of us are interested in new tools and networking. We do have plenty of seats. For those of you who are participating online, feel free to find a gap in your schedule and come and network with other people. Many of you are coming here to learn about clinical application of AI in medicine. We have great sessions coming up. See what's going on in the field. This is awesome. All right. I'm not gonna close the poll, so continue to enter the information. I would like to show you a little video, if I may, to show you one example, what I felt it's pretty exciting. I can get the network.
To this end, a team of researchers from the Jameel Clinic at the Massachusetts Institute of Technology and Massachusetts General Hospital developed a deep learning algorithm that can predict individual lung cancer risk without clinical or demographic data inputs. The algorithm, named Sybil, uses data from a single LDCT scan to predict the likelihood of the development of lung cancer 1 to 6 years in the future. Sybil was trained using over 28,000 scans with known lung cancer risk and was programmed to learn from expert annotations of biopsy-confirmed cancers to better reason over the entire LDCT. The team evaluated the performance of Sybil using 3 independent data sets of LDCT scans of diverse patient cohorts from the National Lung Screening Trial, Massachusetts General Hospital, and Changgung Memorial Hospital in Taiwan. They found that Sybil could forecast short and long-term lung cancer risk from the LDCT scans accurately for a wide range of patients and on modern scanners. Moreover, it provided relevant information regarding future lung cancer risk in patients. Sybil has the potential to revolutionize lung cancer screening and management, enhance personalized screening, and reduce the overall frequency of biopsies. Combined, these applications can increase cost effectiveness and may make LDCT-based lung cancer screening feasible even in low-resource settings. Importantly, the preliminary data indicate that Sybil may be a powerful tool for prioritizing patients who are at higher risk of lung cancer, regardless of their smoking history. Further research is warranted to enable Sybil for real-world clinical applications.
Well, example like this can be found everywhere. And I'm simply pulling out a recent publication and featured by ASCO. And in fact, this video was made by American Society of Clinical Oncology. And I simply want to show you that AI is real. AI in medicine is coming. And you're going to see more AI in medicine in the research stage and probably in the application in the future. And all of us probably agree that this is a golden age to be in this field. And let me see if I can get to the next page. And the age of AI has come. And I just want to bring out one article that I read from last year. And the AI today is very different from machine learning, even going back 10 years ago. The AI today take multimodality data. It actually can use a few labels to learn, and it will have many, many parameters. And in the case of large language model, could be trillions of parameters, and they run on massive computational power. And many, many opportunities you're going to see today include the precision health, digital clinical trials, hospital at home, disease surveillance, digital twins, virtual health doctor, and so on and so forth. And I think if you are enthusiastic, and I would like you to withhold your enthusiasm. I do have a short video, but I don't think I have a time. I would like to play it over the lunchtime and then give you some treat. So without further ado, I would like, this is a video. I would like to introduce our keynote speaker today, Dr. Trey Ideker. I've known Dr. Trey Ideker for more than 20 years. He's currently a professor of medicine, bioengineering, and computer science at UCSD. He's a co-director of UCSD's PhD program in bioinformatics and systems biology. He's a director of the NIH Bridge 2 AI Functional Genomic Program, National Resource for Network Biology Center Director, and many other NIH and NCI Center Directors related to CellMap. And he is certainly well educated. And many of you may not know, but I know I personally attended the lecture by Lee Hood, one of the founding father for systems biology with Trey, and DNA sequencer inventor. And Trey was his PhD student. And I believe that Trey went to MIT Whitehead for his postdoc. He's editorial on the editorial board of so many high impact journals, including Cell, Nature, and most impressively, he was achieving all of these accomplishments when he was really young. He was decorated as MIT Innovators 35 under 35, and many accomplishments just praised by International Society of Computational Biology. But I would like to point it out that most of you, even if you don't know his many impressive accomplishments, you actually use his software tool and Cytoscape. And one of the things that I think really impressed me was even before I knew Trey or Cytoscape, I read his science publication where he co-authors with Lee Hood that talk about how network could actually help uncover protein function in a landmark science paper. And that's how I got to know his work and watch his career trajectory just exploded. He's one of the person that keep on innovating, keep on finding new pathways to spearhead our field. So without further ado, Trey.
You can do this. Thanks, Jake, for that really nice introduction. And I think while he's getting the slides up, just a word about Cytoscape. I think one key decision we made at the time was to go open source back in, and this was back in the year 2000 or 2001. I wasn't actually originally on the side of open source. I had some idea that okay, it'll be free to academics, but maybe there's a corporate potential with this thing, and I was talked out of it, and it was the best talking out of I think I ever had, because then what happened is a lot of people were comfortable coming in from the community, and over the years there have been probably about a dozen different developer sites involved, and at the moment my group is still involved, but there's heavy involvement from UCSF and Toronto, and in the past European labs as well. Looks like it's almost ready. Okay, thank you very much. So today I'm going to talk about AI applications in cancer, as well as data generation efforts in cancer, and you'll see how it all fits together. Jake already mentioned multimodal data generation and integration, and you'll hear more of that in my talk and how we're essentially hastening towards, I think, a system for precision cancer medicine. But first, the premise or the point of departure for all of this, and that is this longstanding challenge of what's known as genetic heterogeneity. This is still arguably the big unsolved challenge, not just of cancer, but of essentially complex disease genomics. And as you probably know, most common diseases are absolutely complex and multigenic. In fact, far more multigenic than I think was ever appreciated. So I'll talk mostly about applications in precision oncology, but you might be thinking of applications in other diseases as we go through this. So what do I mean by genetic heterogeneity in cancer? People talk about within tumor heterogeneity and between tumor heterogeneity. Here in this slide, I'm focusing mainly on between or patient-to-patient heterogeneity, but the same argument is made for looking from cell to cell or region to region within a single patient's tumor. And it's the simple fact that as you look across these different samples, you will find that there's a few recurrent cancer mutations that always arise, or very, very often arise, I should say, not always, but a much, much larger number of rare somatic mutations. And that principle is often shown by plots like this. This is called a long tail tumor mutation plot. This is for the Cancer Genome Atlas colorectal cancer cohort, published more than 5 years ago now. It had about 500 patients in it, maybe just shy of that. And what you can see is the 4 most frequently mutated genes across that cohort are shown: APC, TP53, KRAS, PIK3CA. But then very quickly you get into this rare gene mutation land, where all of these mutations are observed in fewer than 10% of patients. There's the 10% mark right there. And essentially all of these genes are fewer than 10, and then very quickly fewer than 5% of patients, leading to the question of how we treat these thousands and thousands of genes in that long tail. And I should clarify, this is not at all even looking at the non-coding genome. This is just coding changes in which lead to coding changes in proteins. Well, that question is still a very open challenge in cancer genetics, but I want to point out that at least for some of these genes, we already know that there is a strong role. And so PIK3R1, B2M SMAD3, AXIN1, I've just labeled 4 of the genes out here that have a lot of evidence that are involved in the pathogenesis of colorectal cancer. And the key to everything I'm going to be talking about is already shown here just by looking at the gene names. And what I'm going to talk about is a potential solution to how you can deal and integrate across all of these rare mutations. So you see here, PIK3R1 is the regulatory subunit of PI3K complex, and PIK3CA is the catalytic subunit, or one of them, and R1 is one of the regulatory subunits. SMAD3 is but one subunit of a large transcriptional family of factors that form these SMAD complexes, and that's the one we're seeing here mutated in about 5% of patients. But of course, it goes together with many others. So the idea here, and which is not at all novel, by the way, for decades, people like Bob Weinberg and others have been talking about the hallmarks of cancer, and that is to say the pathways that underlie pathogenesis of this disease. And each pathway is absolutely involving many different genes. So the question isn't that cancer is a pathway-based disease. It's really how to identify what are those pathways, and then once you have that map, which of those pathways are under mutational pressure in a given patient. But the idea would be that what you're really looking at here is not a single gene disease that's driving the disease, but what's really going on is there's a protein complex or signaling circuit or other type pathway that's under pressure. And when you break that apart at just the gene boundaries, you might see that A here is a subunit is rarely mutated, or D. But if you put it all together, the mutation burden on the complex or pathway as a whole can be quite large. Over here on the right is not just an illustration of that principle. It's an actual plot that we've created in the course of this type of research, and I'll get to that as we go on. So now I've changed the x-axis from genes to protein assemblies, or multigenic protein assemblies. And when you look at that in colorectal cancer, same cohort, what you now see is a collection of nearly 100 assemblies that are mutated in 50% or more of individuals. Now, you still have to, not all of those, it turns out, are relevant to the disease, and I'll talk about how to figure that out, but nonetheless, you get the point. You now are no longer dealing with rare events in cancer. So first, though, where is this map of cancer protein complexes and assemblies that might be important for understanding the cancer genome? And that itself is an enormous challenge. So here is another slide, which I am not the first to show, but is very important for the narrative here, which is that, as you might have heard before, the human genome has revealed essentially a list of parts. That is to say, the genes and the gene products, i.e. the proteins that are encoded by that genome, but not how those parts fit together. And this is akin to taking this Mark I Volkswagen Golf here and decomposing it into its component parts on the factory floor, as the VW technicians have done. but then not having the assembly manual shown down here in the lower left for how to put those parts together into the final golf. So in this talk, at least the first, say, half or so of the talk, the goal is really how do you read out in a systematic way what that assembly manual might look like. Well, certainly we have some clues as to what that assembly manual looks like if you go into databases like Protein Databank, PDB, and you look at the beautiful 3D structures of protein complexes there, which of course are multigenic machines already. Proteasome is a classic structure that degrades proteins. It's sort of the garbage collector of the cell, as you may know. Here's a more recently deposited structure during the COVID epidemic, the SARS-CoV-2 RNA polymerase, including the RNA template in that machine. So it's a protein RNA multi-component machine. But despite all of that productive work through structural biology deposited in PDB, we still largely agree, I think, that I mean, it's certainly a arguable topic, but I think many of us would agree that we've just scratched the surface. And so here's an example of a larger organelle-sized machine in the cell for which there is absolutely no structure of any kind. And in fact, we don't even know what are all the proteins that go to the neuronal synapse. There are probably 1,000 or more proteins that are part of the synapse structure. We know maybe 500 or 600 of them. And even for those, for many of them, it's not clear where in that structure those proteins belong and what are their dynamics. And this type of Jetsons-like city of proteins and other molecules flying around is really, I think, going to be the rule rather than exception. And I don't think that's at all controversial. So the question really is how do we get at, in a systematic way, just like the Genome Project systematically read out the structure of the DNA, the rest of the cell biology around that DNA? And so to start to get at this, we put together a project that Jake and I are both involved in, and the opportunity came last year in this NIH Bridge to AI initiative. Just a word about that initiative. It's a common fund. If you know about the funding structure of the NIH, it's a common fund project where multiple NIH institutes contributed support for the multiple projects that were finally funded by this Bridge to AI initiative. These were meant to be, notice it's not AI, it's called Bridge to AI, and the key word there is bridge, and the bridge is the data. So what they want is AI-ready data sets that will then power the next generation, not just of our team, but of the whole community, to build machine learning models. There have been 4 data generation or DGP projects funded. Ours was one. I'll talk about that in the next slide. And the other, ours is a very basic cell biology structural project, as we'll talk about, and then with some allusions to how those mesoscale draft maps of human cell biology can be used in machine learning systems. The other 3 were generating patient phenotypic data from the EHR and like resources. So I think we're also quite unique in that we're the one molecular project that complements the other 3 patient data-centered projects. So here's it's a large collaboration of folks. Here's Jake down here leading the teaming module. I'm a lead of the tool development and optimization module, and then we have data acquisition, standards, ethics, skills and workforce development, and so on. Now the data acquisition and the logic for that Bridge to AI proposal came from a proof of concept developed in 2 earlier applications. One was called the Cancer Cell Map Initiative, and the other funding stream was called Treatment Futures. And here's the sort of subset of folks. It's an overlapping set of folks, I should say, Looks like I've lost in printing a PDF. The names have gotten jumbled, but not too badly. So here are 6 of the faculty from those earlier initiatives: Nevin, Emma, and Prashant, and this is Andre Shali, although his name has disappeared. These 4 faculty are also involved on that last slide in the Bridge to AI initiative. And here's some of the earlier work I'm going to talk about today that I think really has shaped that Bridge to AI initiative. And most important, what I want to show are the trainees who've actually done all the work I'll be talking about here. If this sounds like an acknowledgement slide, I assure you that it absolutely is. So just in case I forget to acknowledge these folks at the end of my talk, and I'll be talking in particular in the first part about some really nice work of Yue Chen. She's now a Schmidt Fellow, talking about a Schmidt Futurist. She's now a Schmidt Fellow at the Broad, and Jinzu Ma, who's now a founding faculty member at the Beijing AI Institute. Okay. And then I'll mention others as we go. So here's what the end-to-end cell mapping and AI platform is meant to look like. We have, and meant is maybe a conservative word. We have put all of this into place in draft form, but I also want to make it clear that I'll certainly make no claims that we have the end-all be-all solution here. This is just where we are now, and I think we have a long way to go. Our cell mapping platform is driven by 2 primary modes of what I'll call mesoscale structural data. But the key thing here, so we're not going to be going after in this draft 3D structural coordinates of every molecular machine we can see inside of a tumor cell. We are going to be going for an ontology of parts that we can see, okay? And then the idea being that we get full coverage of a cell using this sort of coarse mapping technology, and then we can come into particular components that have been discovered with X-ray crystallographic and other structural approaches. Actually, I should say cryo-EM is probably the dominant approach here in terms of structural biology. But let's talk about these coarse mapping techniques. So one is protein-protein interaction mapping by affinity purification tandem mass spectrometry. And that draws these large protein networks and in a particular cell type. And then you mirror those experiments with protein confocal microscopy, or I should say immunofluorescent protein confocal microscopy. You put those 2 together to read out these data-driven ontologies of subcellular components and protein complexes, again, with the idea of getting full cell-wide coverage. And then you put these ontologies into as knowledge bases for AI systems in precision medicine for reading the disease genome towards prediction of therapeutic outcomes. And I'll talk about now for the rest of my talk these 3 different pieces. So first of all, just a word about those 2 input data types. On the left here, we have protein immunofluorescent confocal microscopy. On the right, we have affinity purification mass spectrometry. From a high level, I like to think about these 2 major technologies as basically just a leading edge, not the only leading edge, but a leading edge of 400 years of microscopy and more than 400 years of biochemistry on the right. So what do microscopists like to do? They look at stuff in largely 2 dimensions under microscopes. Here you fluoresce a particular target protein using an antibody against that protein, and that changes from experiment to experiment or image to image across a say 96 or at least 24 well plate. Then you counterstain against a set of common landmarks that never change, that then allow you to triangulate the position of that first protein as it changes. And here you're looking at so green is the target protein, blue is staining the nucleus, and red is staining microtubules. What's not shown here is you often have a cytoplasmic membrane and sometimes a cytoplasm stain as well. On the right here, and so each of those images is giving you the distribution within the cell of a given protein. what do biochemists do? Biochemists don't like microscopes. They like to grind stuff up and pour it over columns. And that's exactly what's done here. But you still have an antibody against your target protein in biochemical parlance. That's called the bait. And then you are isolating that bait along with whatever proteins it is thought to proximally interact with, called preys, and those are identified using tandem mass spectrometry in terms of their identity. And of course, a control there is you should also re-identify the bait. And having done that, you draw these protein interaction networks. Now, what Yue Chen and my group realized about third year into her graduate program is that these two approaches, although they had never been formally put together. Now, actually, I should back up and say, very often you had a very large protein imaging study where they used a few APMS pull downs to validate their results, or vice versa. You'd see a few images in a very large APMS study. But in terms of actual front end loading of both data modalities, that had never been done. And I think one of the reasons it hadn't been done is it just wasn't clear what the sense in doing that would have been. Why would you do that? Well, what Yue realized is that, in fact, these are two ways of positioning proteins. One is more global relative to the whole cell, and the other is very local relative to your neighboring proteins in whatever machine you're in, or machines, okay? And so combining that idea, or taking that idea to the next level, that was a very attractive idea to someone with a machine learning background, and she has both a biological and machine learning background, and that was what she was hoping to achieve in her PhD. Because when you think about embedding data types using machine learning models, you're essentially positioning whatever objects. Here, the objects are proteins, but if you're doing, say, single cell transcriptomics, the objects are cells, what have you. You're positioning, or patients, if you're analyzing the emergency medical record, the EHR, electronic health record, you're positioning patients in that embedding. But here it's proteins, and we simply want to position proteins in that embedding via two ways: one via images and one via protein networks. If the word embedding is foreign to you, I can say two things. One, much machine learning research has been devoted to coming up with what is a quote unquote good embedding, meant to be a simpler or lower dimensional representation of a complex input data set. And so if you're embedding, our embedding here, it turns out has 1,024 dimensions, which sounds like a lot, but it's a drop in the bucket compared to the number of dimensions you would have, number of pixels, say, you would have in a confocal microscopy image, or even in this APMS data set. Now, even if you have not made embeddings or studied embeddings yourself before, if you've ever seen a talk or done research on single cell RNA-seq, you are inundated with embeddings, because every UMAP or TISNE plot you've been shown is an embedding of that complex data set. In that case, the points, as I said, are cells, not proteins. So in our case, they're proteins, and the position of a protein relative to others indicates proximity of protein location in the cell distribution, and proximity in that protein interaction network. And that's verifiable, of course. You can go back for that particular cluster and check by eye that it got it right, okay? Moreover, because these are two structural positioning technologies for proteins, structural being the key word, I can calibrate distance through that embedding to nanometer protein distances of the object that protein cluster forms. And I can do that using Protein Data Bank. And that, I think, is a really unique thing we've been able to do that you can't do in, say, a cell embedding of single cell RNA-seq, because that's more of a functional embedding, or a cell type embedding. This is absolutely a structural embedding. And then, as I'll show in just a second here, having convinced yourself that that is reasonable, you can then cluster the proteins in this embedding in a hierarchical way to resolve as many subcellular components as you can, spanning multiple size scales, and we'll see that in just a second. Now, we call our thing the multi-scale integrated cell. It is very similar to a paper that came out about 6 months later by the Chan Zuckerberg Initiative people called OpenCell. So just to kind of make you aware of both works, they both started in HEC293 cells, so not a tumor cell type, a human embryonic kidney cell type. And the reason is simply that that's a workhorse cell line you might be familiar with. And we already had a lot of data to get started with thinking about how you build these cell maps. In our case, we collaborated with Emma, who's now, who is one of the leaders of the Human Protein Atlas for the imaging, as has been developed over more than a decade. And on the protein interaction side, Steve Gigi and Wade Harper and Ed Hutland at Harvard, who had developed this protein interaction map called Bioplex, and were beginning to really flesh it out in HEC293 cells. However, those two projects had gone on independently up until that point. So we only had 700 proteins to work with where we had data in both modalities. So everything in this music map for our proof of concept is really just 700 proteins, okay? We are now, through the Bridge to AI project, just about to get to 5,000 proteins, I should say, for a different cell type. But I won't talk about that today. That'll be my talk next year or whenever. So let's get into it. So having come up with those embeddings for those 700 proteins, or for now 5,000 proteins, you now look at cluster structure, and we use an algorithm called that's a class of what's called multi-scale community detection, where you do this clustering over multiple scales. And so you because we can calibrate here to nanometer sizes here, if I can move this down, you can see it better. We're starting at just a very small, several nanometer radius around one protein, and looking at what clusters we see, and we pick up things like GMN7, SNR PB2, and so on. You now raise the radius, and you pick up some more proteins. And now, as you step up to about 25 nanometer range, look at what happens. You now merge one community with another, because of course this clustering procedure at any given scale is happening all over the map simultaneously. And if you look at what you've just done, you've just essentially discovered a hierarchy of subunits inside of what we know and well is the pre-catalytic spliceosome. So we've simply reconstructed the spliceosome via its U1 and U2 subunits. All right, that's what's just happened. And so now, in looking at the whole map, I am going to abstract away the individual proteins and look at just the protein complexes in the middle here in a hierarchy. And that's what's shown over here at the right, because again, I have sizes for everything. I have a size ladder I can put on the right, and then essentially lay out each component that's detected in a vertical top-down divisive manner, or you can also think about it as bottom-up, agglomerative manner. You have the bigger components up here at top, like organelles, and then they factor into smaller and smaller compartments inside of those organelles, and finally down here at the bottom into specific protein complexes that again are resolvable by these two data types. The yellow components are well known. The purple ones are either entirely novel, or more often have enough novelty in terms of the proteins included there that we did not feel comfortable calling them known. Here's just another a slide on the calibration, which I think is important. So now that we have each of these components, we get a predicted diameter of that component in nanometers. And now here we're taking PDB and other literature for components that were not used for the calibration initially to check how well the music can predict that diameter. And notice it's doing a pretty good job. This is a log-log axis, by the way. It's easy to make things look linear on a log-log axis, but nonetheless, I think the point is clear that you're able to capture about 4 to 5 orders of magnitude of scale. Of course, 10,000 nanometers is about the size of a human cell, depending on cell type. And then here's for instance that spliceosome that I showed you the recovery of. Here's what the structure of that looks like in PDB. The measured nanometer diameter of that is 42, and through bootstrapping we can give a size range of our music map for each component, and we estimate its size to be between 26 and 90 nanometers. Okay, so that's that's the first part of my talk in terms of how we get these structures. Now let me talk about application to disease and disease genomics. First, I have to go back and now apply this to cancer cells, because everything I just showed proof of concept in was a HEC293 cell. And I also have to tell you that the imaging side of the cancer story is still developing, and is exactly what Emma Lundberg is now doing. And some of us just spent Monday at NIH talking about making more plans around and so on. But what we have so far is a interesting map of the other side of the equation, protein-protein interactions, across now a growing cadre of tumor cell types, lines and types. What we've done so far, and this is now with Nevin Krogan, who is a mass spectrometry expert at UCSF. We have tagged 61 different frequently mutated cancer proteins across multiple cancer types. So if you think about that long tail again, we're going to start by exploring the most frequently mutated proteins or genes at the beginning of that, and then work outwards from there. But every time you pull down a protein, you get an unbiased, or we're certainly biasing it by that bait. But then around that bait, you get an unbiased view as to what are the preys or the interacting proteins proteome wide. And so this map isn't just restricted to 61 baits, but it extends to nearly every protein encoded by the genome. By the time you're done, and then I'll show you in a second here. We then backstop still with existing protein interaction data in the public domain, which is largely not cancer specific, but nonetheless sometimes it's important to see to connect the dots. We've done this mapping across 6 different tumor cell types, covering breast and head and neck cell types. And then this says in review, this actually just got accepted. We have another 22, our map of 22 proteins focusing on the cancer, of course, all important process to cancer DNA damage repair, and that adds a dynamic aspect of DNA damage. So here's an example shown here in the middle of integrating new and existing data. PIK3CA got pulled down and identified through the blue interactions here, these interacting partners. And then in the other colors are existing data from the public domain that show you that those partners, in fact, themselves are a protein complex, which is actomyosin, okay, or at least one version of actomyosin. And the novel complex here isn't having recapitulated actomyosin, it's that PIK3CA, an infamous cancer driver, interacts with that physically. Through further functional studies with Sylvia Gutkin's lab, we can show that you can drug PIK3CA or myosin and show synergy between those compounds, and that synergy can be pushed upstream in terms of the molecular pathway, because you also see it in sort of proximal downstream readouts like AKT phosphorylation. But so that's in this paper, in these papers down here. But what I really want to focus on is back to this issue of mutational analysis. So what you've also done by forming this complex is you've put together a commonly mutated cancer protein, PIK3CA, which in the case of head and neck cancer is mutated in about 18% of patients, with more rare mutations to actomyosin proteins, but that add up. Okay, and so the total mutational burden on this complex is about 30% of oropharyngeal cancer. Here's an example of a known complex. So that last one was a novel complex with PIK3CA. Here's an example of a known complex where we've simply reconstituted collagen. Okay, congrats to us. We got that one. But what was not well appreciated at all was the fact that collagen complexes are under intense mutational pressure in multiple tumor types. So what's collagen? Just to make sure we're on the same page, it's the dominant factor of the extracellular matrix. Collagen is formed by these triple alpha helices of 3 collagen subunits each, drawn from a much larger family of collagens. And then that braid then rebraids with others to form these microfibrils and fibrils, and finally the collagenous fiber. Collagens had never been observed to be under significant mutational pressure because each one is rarely mutated. So this is showing the distribution of mutation frequencies across the collagen family in lung adenocarcinoma. You might have thought that 21% of patients mutating collagen 11A1 would have had a pulse on that radar, and it turns out, much to my surprise, when I looked at this, they did not report collagen 11A1 in the lung adenocarcinoma TCGA paper. And they didn't because lung cancer has one of the largest mutation burdens of any cancer type. And so every gene gets a healthy number of mutations genome-wide, and this one simply was buried in the long tail, strangely enough, okay? But then when I discovered that half of lung cancer, 49% of lung adenocarcinoma is mutating collagen, that becomes more interesting. You might be thinking, well, Trey, the bigger my umbrella, of course I'm going to catch more patients. And so you have behind the scenes here statistical tests, much like the original cancer genome projects run at the gene level, now at the multi-gene set level, to make sure that I have significant mutation pressure. What's in this paper, but I'm not showing you here, is there's other cancer types where the mutation burden is even higher. Melanoma, more than 70% of melanoma patients are mutating collagen. Again, no one's ever found collagen mutations before, shockingly, because they never looked at the level of the assembly. And then with Stephanie Fraley's lab, what we're showing here is we can create mutant matrix and compare it to normal matrix for selected mutations found in these cancer cohorts, and watch the behavior of cells seeded into that matrix. We thought cell migration was going to be affected. It was not. What's affected like gangbusters is cell proliferation, as you can see here with this Ki67 marker in the mutant collagen matrix. These cells are proliferating to a much higher degree than in the normal matrix. So that's two examples, but of course, as I showed for the music map from Chen, and this, by the way, is the work of Fan Zhang and Mark Kelly in my group. Of course, Fan has applied similar cluster and community detection approaches all over the map to get a hierarchy that we call nested systems and tumors. Fan thinks about, I think Yua started with the cell at the top. Fan started with the cell at the bottom. So the whole thing is rotated 180 degrees. And so now you can think about the whole set of proteins in tumors is down here, and then you're factoring them into smaller and smaller components moving up the page. And then what his co-author Mark Kelly did is simply make, oh, this was supposed to be an animation, but I realized I printed a PDF. Never mind. But this, for instance, takes one cancer type. It's going to run through all 12 if you let it. But this just shows the particular mutation pressures in one disease, bladder cancer, on this map of protein complexes. Of course, every cancer type mutates somewhere in the map, and that's why the root is black and the large hallmarks are black. Every bladder cancer has to mutate cell cycle, okay? But how does it do it? That's what you see as you move up the hierarchy. I mentioned that we had in Press this cell system, or in cell systems in Press this the next map. So I'm not going to talk about it here in interest of time, but please do check out this map we call DDREM, which stands for DNA Damage Response Assemblies map. Similar idea, but now pushing very specifically into an effort to define the protein complexes in this kind of mesoscale draft way that one can see among DNA repair proteins, including signaling of repair. What I also I'm going to use this slide to show is we're thinking a lot in these projects about visualization. So you can show this map hierarchically in terms of like an ontology of parts and cells that you can resolve via this data. Another way to look at it, and I think there's uses of both types of visualizations, is in a very as a nested set of protein assemblies or circles. And that's what's shown here. So, for instance, here you're looking in this highlighted region at double-strand lesion repair, or our version of that through the systematic, as it's been reconstructed via the systematic mapping approach. There are the underlying protein connections that formed, that complex in the underlying data, and it's shown over here at left. in these nested circle packing diagrams. And so there's the complex. If you were to look inside it, it's not simply a uniform set of proteins, but there's substructure that we've labeled here, homologous recombination on the left. That's this yellow cluster over here versus non-homologous end joining over here, complex on the right. And I think that maybe illustrates the idea well enough. And so you're kind of looking at this kaleidoscope of objects using these circle packing diagrams, which we think has some promise. But in the last 10 minutes or so of my talk, what I want to do is now talk about the last piece of that end-to-end pipeline I showed at the beginning for how we think these data-driven maps of tumor and other disease cell biology can be used to interpret genetics, interpret the human genome. With a view to clinical translatability. So you'll see how we get back to those maps in a second, but let me first just start fresh and let's talk about AI and ML in precision medicine. I think as Jake just alluded to in his talk and others will in their talk, I think we're here today essentially because AI has made fantastic strides in multiple areas of biomedical discovery and clinical application. The one I'm going to talk about in the last few slides here is specifically predicting the response of a cancer patient to therapy given their tumor genome sequence, or at least a gene panel, as is now commonly measured and paid for by insurance. So that all of these problems start to look like an input-output relation for a machine learning model. And again, there's variations on this theme, of course, but in my case, the input is going to be a patient genome or most simply a list of mutated genes in that patient's tumor. And then you perhaps you have another input here, which is what drug you're thinking of giving. And then what you'd like it to do is simply say yes or no, responder, non-responder. Right? Good idea, bad idea. That's one simple incarnation of precision medicine. There's been a lot of concern and talk these days about the so-called black box problem with these modes of patient diagnosis and treatment. And the problem is that these machine learning models typically are trained to optimize predictive accuracy. How well do you predict a patient's response to a particular drug? Okay, but what's not at all treated in the modeling is why. Okay, what in the tumor cell or in the tumor tissue or is actually causing a patient or causing me to predict as the model that patient's going to be non-responsive. And so this is a big problem in machine learning that's well appreciated, called the lack of interpretability. And by the way, there's a closely related problem known as lack of transferability, where you cannot explain, you often cannot translate or transfer to a different cohort outside of what's been used for model training. So how do we deal with this problem? And there's a number of efforts out there, but here, the specific progress we've made is by using those cell maps I just spent the first 40 minutes talking about to structure the inner workings of deep learning models for precision medicine. And so now I think, permit me, I've rotated the genotype-phenotype equation top down, okay? So there's your genotype plus drug. You want to predict, in the case of tumor biology, simply how fast is the tumor going to grow? So something akin to a proliferative phenotype in its simplest form. But now, instead of simply using an out-of-the-box or I should say in the box deep learning artificial neural network, I'm going to specifically assign banks of artificial neurons in there to each subcellular component that's been charted in that map. So for instance, back to the DNA repair example, maybe you'll have in there a component at a high level that's just representing DNA repair, but then as you factor that down into the subcomplexes, you see there's a complex related to double strand break repair, another alternative repair mechanism, base excision repair, and so on. And all of those, each of those components has a bank of neurons assigned to represent the functional state of that component under a patient's tumor genotype or list of mutations. Okay, How do you train these models? We start in cell lines where all the data are. Okay, it's not the most relevant data, but it's where we have the most data. There's no question. And then we translate, if we have PDX models for that drug and cancer type, we'll then translate first to PDXs and then finally to patients. The place we usually start, again, is cell lines, and we have, between work of the Sanger Center and the Broad over the past decade, a large matrix of about 1,000 different tumor cell lines, each of which has been fully sequenced. So we know the mutations, and each of which has been exposed to actually now more than about 1,000 different compounds. So that's 1 million different data points if you want to do them right. It's simple math there. And it's actually not quite 1 million because you miss some combinations, drop out, but nonetheless, it's a lot of training data. And so that's essential. And then for each of those combos, cell line cross drug, you have a dose response curve that you try to predict and then translate to patients. And so, in interest of time, because I only have another slide or two to times worth of of material here, I'm just going to go right to a result. This is an unpublished result that I'm excited to present. Again, I see there's been some loss in the PDF translation process. I apologize about that, but you'll get the idea here. So we had a model that we've trained to predict the response of breast cancer patients to inhibition of cell cycle kinases, CDK4/6. Palbociclib is one that's in the clinic, and it's got 2 others: ribociclib, abemaciclib. These are all inhibiting CDK4/6. Slightly over half of patients don't respond, but nonetheless, many patients do, and you'd like to tell which is which, of course. So we first trained this thing in cell lines, and we got a very, very high predictive accuracy, which is why I'm talking about this drug or this class of drugs and not some other class of drugs. But for about 1/3 of the drugs we've looked at, you get a pretty good predictive power. Okay, so this is not cherry picked. We had about 1/3 of the drugs to choose from here, but clearly CDK4/6i is an area of active interest. This is the true area under dose response curve of each of the cell lines. And in yellow is the model's prediction. So it's getting mostly right that these are all resistant tumor cell lines in yellow, and mostly right that these are all sensitive. Odds ratio of 40 is just screaming. And I said, there's just no way we're going to see an odds ratio of 40 in patients. And we don't, but you'll see it's not bad. So then we go to PDXs. I'm just going to go clockwise around the screen. Here's the PDX result. We also got a decent result in PDXs. Here we didn't have to generate any new mice. This is you now leveraging this nice resource, the PDX Encyclopedia. And then here we leveraged Project Genie to do a retrospective clinical trial on response to CDK4/6i. These aren't all palbo that was used here for these patients. It's a mixture of palbociclib and ribociclib. And back to the EHR, their EHR didn't actually say whether it was palbo or ribo. It just says CDK4/6i. Okay, I think our model might do better if I knew which was which. Just saying. Okay, but nonetheless, you can see here that for patients who get a CDK4/6 inhibitor plus standard of care endocrine therapy, and then you look at how the model stratifies patients. It hasn't seen the outcomes, of course, the survival outcomes, but here's in blue is the patients the model predicted would be sensitive to that, and then of the resistant patients, we thresholded in 2 different ways to get a partially resistant and a fully resistant cohort. That's still a pretty good hazard ratio. And then over here on the right is the control, where among patients who did not get a CDK4/6i, let's still let the model stratify them into CDK4/6i resistant and non-resistant subgroups. And you should not see a separation because these patients haven't gotten palbo, and in fact, you don't. Okay, so now in the last slide or two, the more interesting thing, having now convinced ourselves that the model is accurate, and by the way, there's other models that also get decent predictive accuracy out there, not just ours. But what we can do is open up the hood and ask the model mechanistically why. what are the pathways through protein components in this pathway assemblies, I should say, that are most important for integrating mutations in the genotype to predict palbociclib resistance or sensitivity. And here is that nested systems and tumors map yet again. Looks a little different, but it's the same map. And in red and yellow, in the colors, are those most important pathways. It's good as a positive control. Here is the CDK4/6 holoenzyme. That's what the actual network that was reconstructed looks like. So it's not just CDK4/6. There's that component, but it's the upstream signaling inhibitor, CDKN2A, and then some downstream stuff like RB1. But that's the complex, quote unquote, that gets pulled out of the high throughput systematic analysis. And this thing is clearly or I should say mutations to this component clearly modulate your response to CDK inhibitions as expected. But then what else do you find? And you see a lot of other interesting pathways, some of which have not been well appreciated. And so here's one that we're following up on now. And I should say the first thing we do for those is, rather than trust the model and its explanations, so things like, AKT activin signaling, that's actually, there's a lot of literature on that. Same thing with EGF, FGF receptor complexes, but what about PML bodies? What's that have to do with palbociclib response, and so on and so forth. And so the first thing we do is a CRISPR screen, palbo times gene knockouts, or CRISPR-A to amplify genes in the case that the genetic perturbation in patients is a copy number amplification, to see whether we actually see an interaction with the drug when you do, in a directed way, perturb those systems. And in fact, when you perturb those systems with palbo, you do see an increase in fitness compared to of the cell lines compared to perturbations to control assemblies. And here's just one example of a transcription-related complex that is quite interesting, we think, and we're following up more about. So with that, I'll conclude. I started by talking about a structural biology way to get sort of mesoscale draft maps of human cell types, which is based on protein confocal imaging plus protein affinity purification mass spectrometry. One point I didn't make, but needs to be made here is, with imaging, it had long been thought that the only way to measure protein distance between a pair of proteins was to tag them both and use a technology like FRET to measure their distance or their proximity. But here it turns out with machine learning, you don't need to do that. You simply analyze the images to embed each protein in this UMAP, and then what it's actually learning, just like your eye, when you look under a microscope and say, oh, that protein is in nuclear speckles, and here's another image where that protein was in nuclear speckles. I wonder if they interact. That's what that machine learning system is doing in a linear way, protein by protein. So it's not quadratic like FRET, where you tag pairs of proteins. It's linear. We combine that with protein networks to build these maps of components and cells. And then in the last few minutes here, I talked about how those maps get used to drive or can get used to drive these interpretable deep neural networks for the clinic. So thank you very much, and I'll be happy to take questions. Are we so we are?
Hello?
Thank you. There we go. Okay. Now we have two mics. So the question was, are we reproducing the imaging data? Essentially, was that the question? Well, so exactly. In fact, when I talk about this project, it was a collaboration with originally Human Protein Atlas, Emma Lundberg, and she's now, no accident, she's part of the Bridge to AI collaboration. And that was, our initial work was exactly as you suggest, based on the fact she had already run through an entire proteome worth of protein locations in essentially a couple different cell types. And then she's looking at, well, what am I going to do next? Okay, and so we, if I get up here and I, wave my arms and say we enough, what I mean is a collaboration of people. And the difference between that initial proof of concept and what we're doing now is the images are generated as part of the project as opposed to pre-existing. Now, how do I, having observed a component, can I go back to the images and regenerate them? Maybe is a question. First, I should give you some detail that every protein is positioned based on 2 to 5 antibodies per protein, and HPA takes a little bit of flack because it's a polyclonal, and these are all polyclonal, not monoclonal antibodies, but nonetheless, from my perspective, I've got at least 2 shots on goal for every protein, and sometimes up to 4 or 5 shots on goal. And if I see those different antibodies largely produce the same position for that protein in the embedding, I'm more comfortable, and I can actually reject an outlier if I need to. So that's one way that we kind of build in redundancy. You of course can go back and re-image to check, and we've done that for a small number of proteins. And the other thing we can do is an approach that's essentially a bootstrapping or jackknifing like approach, where we can leave out some fraction of images from the reconstruction and then go back and check how well well, first of all, that's going to change the list of, or the hierarchy of complexes you see, and you can record how much it changes, how sensitive that hierarchical map is to leaving out data. And you can also do kind of like a cross-val thing where you use the held out data to try to recover. I don't know if that got to your question, but might have orbited around your question.
Wonderful talk. My question is, with all this new technology available, like spatial transcriptomics, you think, integrating that, particularly with the music map, you have 700 protein you said, and look at different cancer, different mutated types, or even small perturbation, how this changes using AI models and things like that.
Yeah, so spatial transcriptomics are really, if you're talking about like single cell RNA-seq, really have skipped over cell biology, and they're talking about tissue structure, right? So here, the talk is really focused on not single cell, but single molecule, or the components within cells. Whereas when I look at a t-SNE map, or for single cell transcriptomics, I've skipped. I'm talking about subtypes of cells and tissues, but I don't peer within cells. So I think this is largely what I'm talking about today, complementary to what a lot of others are doing. And you could almost one day, when you look at these hierarchies, in fact, there's a beautiful paper from Mark Gerstein's lab a number of years ago, where they put subcellular structure together with cell type structure into one of these machine learning models, which is pretty cool. So it's largely complementary. Transcriptomics does get used in other ways, though. So can you peer or can you validate the activity of individual protein complexes here? And we tried using their expression level at the RNA level of those complexes in response to because you've got transcriptomics and proteomics across all these cell lines that we've trained on. So you can say, does that set of proteins, is it differentially expressed by the same patterns of genetics I think it should be? It turns out that works far better at the protein level than it does the RNA level. And it works far better even at the protein level when you're looking at the phosphoprotein level than just the abundance. But happy to take that offline. I don't know, Jake, if you want to, I want to make sure I don't disenfranchise someone here. I didn't get the order.
Hello, thank you very much. It's great to know about all the data that's now out there. On the AI side of this, on the machine learning model, you've described one particular way of embedding that multi-scale network into the machine model. How robust is that to the way that multi-scale model is embedded into the machine learning model? Do you get the same results if you choose a different number of neurons or a different way of structuring that?
We have looked, so one, it's a fantastic question. We need to look at it and others need to look at it more. But we do, one thing we do, so this is not a whole answer, it's a partial answer. We have scanned the number of neurons we assign per system, per assembly, right? How complicated is the state of the ribosome, right? Ribosome is an object in that hierarchy, in that ontology. Should it get a bank of 6 neurons? One, figure more than one. Ribosome isn't just off or on, right? But what about 6 versus 60? So we've scanned that and we try to look at where the where the knee is in the curve of performance. Performance does degrade, but we try to sort of look at where it plateaus. I'm not wholly satisfied with that approach.
Okay, thank you. Very fantastic talk. In addition to the protein-protein binding, would there be enough data to also map the what RNA and DNA, those protein band? I think that can be very useful for epigenetic drug discovery.
If I understand you correct, you're suggesting that we should have protein DNA and protein RNA interactions in that map, and I could not agree more. I think we're probably out of time. So
Yes.
thanks again.
Well, Trey, as always, amazing science, and this is going to be so exciting when it can be put into practice. Thank you so much.